Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Vladimir Teif from the University of Essex on this show. Vladimir, please let me briefly introduce you to our audience. You got your PhD in 2004 from CEA Saclay in a joint program of the French Commission for Atomic Energy and the Belarus National Academy of Sciences. You then stayed at the Belarus National Academy of Sciences until 2010 while working in the US, Israel and France. You then moved to the DKFZ and the BioQuant in Heidelberg to work with Carsten Rippe and also doing Biomos postdoc. And then in 2015, you moved to the University of Essex, where you first were a lecturer and now you are a senior lecturer at the School of Life Sciences. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
Hi, so first of all, thank you very much for inviting me. Hello, everyone who is listening. Well, how I became interested. As a child, I was dreaming of becoming a medical doctor and other heroic professions that children dream of, to help people. My grandmother was a medical doctor, but somehow I became a scientist. When it was time for me to go to the university, my mom predicted, and rightly, that the future of high tech will be at the intersection of computers and biology. And I also liked physics. So I ended up studying biophysics. Then I was working in very physical and very chemical places until I ended up in the German Cancer Research Center. And even then, I was first working on very fundamental questions. I was actually very careful not to enter the cancer research until I felt that I'm actually ready to contribute meaningfully, know enough about the medical side. And from about 2016, in our lab, we work on liquid biopsies. This is already directly relevant to medical diagnostics. And that's when I felt already that I know enough about cancer research.
So let's talk. Go ahead.
So I just wanted, so it kind of went in the loop kind of completed that at the end I probably doing something that I wanted to do as a child to help people in medical science.
So coming to a science that centers around gene regulation, the nucleosome and nucleosome positioning in differentiation and cancer, I want to start when you were part of Carsten Rippe's lab, because I feel that is when your current research interest kind of started. The first publication then was in AR, looking for predicting nucleosome positions. Please talk about how you got started with Carsten and then the results of this study.
Yes. So maybe one step back after I finished PhD, and my PhD was about a system with very few components, DNA condensation by polyamines. So I wanted to go to more complex systems. I became interested in a transcription factor binding, and there was some period when I was working individually still in Belarus because of my family situation. I had to take care of my family who were ill. And so at that time I was traveling to some nice laboratories, and then the next obvious step is to include, of course, nucleosomes. At one of the conferences, or it was a summer school, I met Carsten. I liked him a lot. And then I came to his lab, first for 1 month, then for half a year. And then I ended up staying in his lab for almost 7 years.
Oh, okay.
And so the first paper in nucleic acids research that you mentioned, basically it was an attempt to make the theory that predicts from DNA sequence, from differential transcription factor binding, and from active chromatin remodeling, how nucleosomes would be positioned, given that you know the rules of how each type of remodelers would move nucleosomes to the left, to the right, or evicted. And so that theory is out there. Well, the problem at that time was, of course, that, and even now, we still don't know these rules, the experimental rules of remodeler action. So therefore, the next step obviously was to directly measure nucleosome positioning in different conditions.
Yeah, exactly. That was my next question, that you looked at nucleosome positioning during embryonic development in that case. And you already touched upon it. So what did you learn from this about nucleosome positioning during this process?
Yeah, so that was the first system that we looked at. We published, it was at that time the first map of nucleosome positioning in mouse embryonic stem cells. And we compared it with neural progenitor cells derived from the same stem cells and with mouse embryonic fibroblasts. So basically, you have three types of cells. You have three types of high-resolution nucleosome maps. Then you look what is different. So the differences are at the level of individual nucleosomes repositioning at transcription factor binding sites, at a bit high level, nucleosomes repositioning at chromatin domains of different type, and also globally, an integral property of chromatin, the spacing between nucleosomes changed. So as the cells differentiated, in this system at least, the spacing between nucleosomes became larger.
And what about the positions? I mean, you talked about the spacing, that it got larger, but did you also, I mean, there is like a very specified nucleosome right at the transcription start site. Did you see any changes there?
Yes, of course. And of course, this changes were specific to those transcription programs that start in the specialized cell types.
Then when you started your own lab, you looked at the nucleosome repeat length. So yeah, you did your postdoc with Carsten Rippe, and then you looked probably for starting your own lab. And how did you end up in Essex?
Okay, so this more than two questions. So I'll start probably from the scientific part. So scientifically, as I mentioned, one of the very interesting things that we found from the first story of stem cell differentiation was the spacing between nucleosomes changed. And I was interested to go much deeper into this. And so this parameter, the nucleosome repeat length, it is defined as the distance between neighboring nucleosomes. Okay, the genomic distance in terms of the number of base pairs along the genomic coordinate between the centers of neighboring nucleosomes. And you can define this globally or locally. So if you have high resolution, you can define this parameter even in a small window of a few hundred base pairs or a few thousand. All right. So, and then we started looking at this in depth. Now, the second was about how I ended up in Essex. Okay, that's an interesting story. So when I was in Germany, I thought I don't speak German. And later, when I already worked in the UK for some time, I realized I can actually speak German. But at that time in Germany, I thought my German is not good. So that was one of the reasons I thought UK might be easier for me to, other than that, the KFZ and Carsten's lab was, of course, very excellent a place. And yeah, I got actually a few offers at that time, and somehow, Essex was selected. Let's make a short story.
Yeah, okay.
Yeah.
What you then did is looking at connections between nucleosome repositioning and DNA methylation as well as CTCF. What did you find there?
Yeah, so there were several kind of stories and papers in that direction. One story was what we call the DNA methylation hydroxymethylation switch. So basically, it appears that DNA methylation affects the stability of nucleosomes. All right, so when the nucleosome sits on a region of DNA that is methylated, it makes the nucleosome more stable. And the antagonist of DNA methylation is hydroxymethylation. There are a few other modifications of DNA, of course, but if you just look at these two, so methylation makes nucleosomes more stable, and hydroxymethylation makes nucleosomes less stable, more fragile. So
why is that? Is that because of the charge, or what is it?
No, it's more probably just structural details.
Because hydroxymethylcytosine is larger?
That might contribute as well, yes. So at first, our result was just the fact that it is like this, the correlation. And then in another paper on this subject in 2019, we looked in wild type stem cells and in cells where there was knockout of enzyme TET, which is inserts in hydroxymethylation. So those cells didn't have hydroxymethylation. So you have cells which have hydroxymethylation and don't have hydroxymethylation, and methylation, of course, is affected as well. And that hypothesis was then directly confirmed in this second study. Okay, this is one line of this interplay, and how CTCF comes into picture. So CTCF binds about 20 nucleotides. It's a relatively it's very large motif for a typical transcription factor. It's large. Okay. However, it's relatively small if you think that if you want to explain differential CTCF binding by changes in the methylation, it's relatively small. Okay. Many CTCF sites wouldn't be regulated by direct change of the methylation in this region of 20 nucleotides. But if you now combine this with this effect of methylation, hydroxymethylation switch that makes nucleosomes more stable or less stable, then you can expand this range. So the nucleosome is 147 nucleotides. So then it's not 20 nucleotides, it's already 147. So you expand the range. So if you change methylation, the change between methylation and hydroxymethylation, also formulation we showed as well. So this switch changes the stability of nucleosome, and then the nucleosome, it was, if it was covering the site of CTCF, or if CTCF was covering the position where the nucleosome wanted to be, then it expands this range of interactions and provides the feedback between these three players. So methylation, hydroxymethylation, nucleosomes, and CTCF. So
does it mean that you have like CTCF sitting there and then a nucleosome comes in just next between the CTCF, or is it like one or the other?
Usually it's one or the other because CTCF binds very strongly.
And would then the nucleosome be, so you just said it, right? CTCF binds stronger than a nucleosome. So if you have a nucleosome covering more space, but a CTCF covering less space, but more tightly, right?
Yeah, so by changing the methylation, you can change the stability of the nucleosome. If the nucleosome is more stable, it outcompetes CTCF. If it is less stable, CTCF outcompetes nucleosome. So this is this interplay. And then on top of that, there is a very long-range effect. The CTCF, whether it's bound or not, by itself, it forms a boundary that then tells whether the methylation can propagate or not, because the methylation attempts to propagate, to spread, and CTCF can actually, we showed, can create boundaries for such spreading. So this provides also an even much longer range feedback.
And would also like be a platform for other factors like coenzyme and things like that.
Yes.
Yeah.
Exactly. Yes.
DNA methylation is also important in heterochromatin, and you also investigated heterochromatin nanodomains and their formation and/or extension. So what are those nanodomains, those heterochromatin nanodomains, and what did you find about their regulation?
Yeah, so what are these heterochromatin nanodomains? So there are regions where you would find nucleosomes packed a bit differently. You can call them nucleosome clutches. In some papers, they are referred like this. We call them chromatin nanodomains. So we're talking about very small regions, like about 5-7 nucleosomes. They're more tightly packed, or generally speaking, they are differently packed than the other surrounding nucleosomes. And so how this Right. And how this heterochromatin nanodomains, they are also associated with DNA methylation, with heterochromatin protein 1, at least those classes that we considered, and we considered several classes of them. So, and we could predict how they are formed. Based on the DNA sequence, you can find some DNA sequence repeats which contain the motifs which are recognized by transcription factors, which can initiate such chromatin nanodomains. And we could also predict where these chromatin nanodomains would end. So where are the borders? One reason for the borders to appear would be, for example, CTCF. Well, literally, it is CTCF, one of the main players, but also some other players who can fix the border at a specific location. But also biophysically, you can consider this nanodomain as a droplet, and you can predict what is the size of this droplet from also physical interplay of forces. Like you have a droplet on a frying pan, and it will not cover the whole pan. So surface tension, so things like in analogy to that. Yeah. So, and we created a theory how to predict this heterochromatin nanodomains. Well, you take into account DNA sequence, concentrations of proteins such as HP1, and the maps of DNA methylation. Yeah.
And how large are those domains in terms of like linear DNA sequence and also like in 3D space?
In 3D space, we actually we made some estimates there in the paper. It was in Nature Communications in 2022, I think. But we actually wanted to connect with direct experiment in 3D, but didn't. That still might be something interesting to do. But in 1D, as I said, 5-7 nucleosomes. Mm-hmm. So this kind of range.
More recently, you looked at nucleosome repositioning or reorganization. The first paper was published in 2016, looking at nucleosome repositioning during differentiation of a human myeloid leukemia cell line, and two other papers focus on nucleosome repositioning in CLL and breast cancer. Along with that, you also developed a script and a database of nucleosome positioning. Since this is quite a large topic, maybe we can kind of summarize this together in this space. What did you find about nucleosome positioning in those cancers?
Okay, so indeed, it's a huge topic. A short summary is that the fundamental question, of course, how nucleosomes reposition in different systems, but there was additional motivation from there that how can you use nucleosome repositioning for clinical purposes? So this is where my second line of research, clinically facing, starts. And if you are working with blood cancer, then, of course, the analysis that you do is you take some blood and analyze it. And there are many ways you can look at DNA methylation, for example, and nucleosome positioning is one of them that we showed. For the first time, we showed that in a system of chronic lymphocytic leukemia, if you just take the nucleosome positioning maps, you can predict for a given sample whether it belongs to healthy or cancer. And also for cancer samples, you can predict whether it is less aggressive or more aggressive type of cancer. And actually, it was a stratification which worked better than DNA methylation and better than gene expression based analysis, because nucleosomes probably predispose events which are not yet happening. They could predispose some cancer program. So that is blood, okay? Now, and then you mentioned also some other cancers. So the bigger motivation then, or the next motivation, one could say, is, so how can you apply it to areas where nucleosome repositioning becomes the main available analysis type, okay? So consider a solid tumor, like a brain tumor, Well, the way how you do diagnostics, you take a piece of tissue and it is called a tissue biopsy or breast cancer. So it's a very invasive procedure. It's better to avoid it. And so it is now becoming very popular, the strategy to do liquid biopsies. So you take instead blood. From blood, you can extract what is called cell-free DNA. And cell-free DNA, this is the DNA that comes from dead cells. In a healthy organism, there are always cells that are dying all the time, and in a disease as well. And so always in some body fluids, there are these pieces of DNA that are called cell-free DNA. In blood plasma, in urine, any body fluid. Now, the connection to nucleosome positioning is very interesting. It's like you are doing the nucleosome positioning experiment in a test tube, but now nature did it for you. And did it in a more consistent way, by the way. So when the cells die, the nucleases cut DNA into pieces, and these pieces go into body fluids. But these nucleases essentially cut between nucleosomes. So these pieces, they reflect the positions of nucleosomes, how they were sitting in the cells when the cells were alive. And then you can do the analysis of nucleosome positioning. So that's how this second line of my research started, with this motivation that you can now apply what you're learning in nucleosome positioning in cells, apply it to nucleosome positioning in cell-free DNA, and that is already directly clinically relevant.
Because you don't need a complicated assay, because you just in air quotes, take the cell-free DNA, because that would represent the positions where the nucleosomes were, and then you just sequence that, and you have like the whole spectrum of nucleosome positions. Is that correct?
Yes, exactly.
Yeah,
exactly. And even better than other methods, because what would you do? What alternative you could do? And that is historically the first thing that people did with liquid biopsies. So you could try to look for mutations. All right. So you extract all this DNA. It's very simple. You centrifuge and extract cell-free DNA from blood plasma. Then, if it's an advanced stage of a disease, there is a large amount of pieces of DNA with mutation that come from these cells with disease. But at very early stages, it's a very small amount of such pieces with mutations, and it's very difficult to detect them.
Yeah.
Especially if you're looking for a limited number of mutations. But nucleosomes, there are hundreds of millions of nucleosomes in the human genome. Not all of them are equally important for diagnostics, but we're talking about many thousands that are important, and their positions change much faster than changes of DNA mutations or, say, methylation as well. So it's a much more sensitive tool.
Yeah. So next you developed a database, right? I also mentioned that. And did that mean that you did a bunch of experiments and took a lot of samples, and then you dropped them into the database, and then you were able to publish that, and people now can reference this?
Yes. When we became interested in this medical side, then I asked, so where are all of these data sets that we can look at? And I started finding some of them and using them for the analysis. Also, even in cells as well, even before looking at cell-free DNA. And so one can spend a lot of time looking for this, and that's what I was doing. Then around 2016, we released the first version of a database which contained already several thousand of nucleosome positioning datasets. So you can imagine several thousand of nucleosome positioning datasets. That's a lot.
Yeah.
And now we're talking about tens of thousands available datasets by now already. And this is the largest repository of this kind of data. And you can do very, very advanced things when you have such a large statistics.
Yeah. I guess so. Was there anything that stands out?
Yes, of course. So, one of the projects that we did, for example, the aging project.
Mm-hmm. Yeah, this was anyway my next question. Like cfDNA nucleosomics, I guess, was the term you coined there.
Yes. Okay, so cfDNA nucleosomics, yes. So
Yeah.
cfDNA nucleosomics, we started applying it to cancer questions, of course, and we developed the technology that allows to do diagnostics based on cell-free DNA nucleosomics. So you reconstruct nucleosome positioning based on cell-free DNA, and then you can detect early stages of cancer. You can stratify patients, and you can do monitoring how the patient responds to drugs. So we were doing this cancer research, and then one of our students came to me and said, "This is an interesting data set." They looked not on cancer, but healthy people, but of different ages.
Mm-hmm.
And it's a small data set. Well, okay, small from the point of view of what we know now, just 12 people, and some of them were 100 years old, some of them were 25 years old, so a very large span of ages. I looked. The nucleosome repeat length increased with age, based on this 12 people. And then that's where the NucPosDB database helped. So we looked at those huge data sets that were generated for the purpose of cancer research. And what happens? People generate maybe hundreds or sometimes dozens of samples from cancer patients, sometimes different types of cancer in the same data set. And they include sometimes comparable numbers of healthy people as a control. And usually these are only used as control. So they are basically discarded. They're just used as a control. Yeah. Like you use input for ChIP-seq as control. But we specifically collected those healthy samples, and there was metadata available with ages, and that gave us a very good statistics. So not 12 people now, but hundreds. And we showed that the nucleosome repeat length increases with age, but now we could show that this is based on a very good statistics and based on cell-free DNA. And not only that, but also we showed that if you reconstruct this nucleosome position in landscapes, then you can, from this cell-free DNA nucleosomics, you can predict age with about 3-4 years precision.
Mm-hmm.
So, and that was the first aging clock based on cell-free DNA nucleosomics.
Oh, well, that's very interesting. Is that what is the benefit of this clock compared to all the other clocks that are around? Is it just another way, or does it show that there are many ways of looking at the same thing?
So there are, of course, historically, the first aging clocks were based on DNA methylation. And more recently, there were other clocks, for example, based on accessibility, like ATAC-seq. So in terms of the precision, the nucleosome positioning is more precise than the ATAC-seq based analysis, because it's with a single base pair resolution. And it's comparable in precision with methylation clocks. Now, you can think methylation is usually a bit more expensive procedure experimentally. And also, usually, to look at methylation, you would need to look at a specific cell type. For example, you would profile a specific or blood cells. So, and you would look in the cells. Yeah? Now, we don't look in the cells. We look at cell-free DNA outside of cells. So, it is complementary. It might become cheaper as well, so which is an advantage.
Yeah.
Yeah, and so it has practical implications.
So if the repeat length increases, this also hints that there are less nucleosomes, right, and less histones when you age. Is that correct?
Not necessarily, but it might be also coupled with this.
Mm-hmm.
So in general, the distances between nucleosomes are not necessarily reflecting the fact that there are less nucleosomes. So these are different things,
Mm-hmm.
but they may actually go in the same direction, these two effects.
Okay. And just in April this year, so we are recording this in 2024, you generated a high-resolution nucleosome map nucleosome maps in paired tumor and normal tissues from the same breast cancer patient using MNase-assisted histone H3 ChIP-seq and compared them with the corresponding cfDNA from blood plasma. So again, maybe verifying the aging clock. So what did you find there?
All right. So this is now in the direction of cancer research. And here we have three signals. So one signal is nucleosome map in a tumor. Okay. Another signal is nucleosome map in a healthy tissue from exactly the same patient, just adjacent to the tumor.
Mm-hmm.
And the third signal is cell-free DNA. So cell-free DNA is obviously the signal that would be later used for diagnostics for liquid biopsy. The fundamental discovery here was that if you compare nucleosome position in tissues, in two paired tissues from the same patient, then at least for breast cancer, because that paper was devoted to breast cancer, you could observe the decrease of the nucleosome repeat length by about 10 base pairs, 7 base pairs, single patient. So it's very precise pair. And that was repeated for several patients. So it's a consistent effect. And that was the first time when it was shown.
Mm-hmm.
So it's fundamentally interesting. This is one. But of course, this is also important for the development of the liquid biopsies, because maybe the nucleosome repeat length is just one parameter, but more generally speaking, the spacing between nucleosomes that you can derive from cell-free DNA. This, as we showed, we have the technology that actually allows to do the analysis of cell-free DNA that gives you information about whether the person has cancer or not, and which type, and how the person progresses. So this is really important.
So when you talk about MNase-assisted histone chip, how does this work? Is this like a standard chip, and then you still use MNase to like digest the chromatin down to like single nucleosomes, or how does this work?
This may be better compared with MNase-seq. So in MNase-seq, you just cut randomly with MNase where it can, and it can only between nucleosomes. Well, it can between the regions that are protected by proteins. And most of the time, this is nucleosomes. So usually from MNase-seq, you do some size selection, and from that you believe that everything that you've profiled is a nucleosome. But if you want to be even more sure, then you could add an additional step, which is immunoprecipitation with an antibody against histone H3, that ensures that those pieces of DNA that you are looking at, they're really those that were part of the nucleosomes and not some other nucleosome size complexes. Yeah.
So imagine you are due to submitting a grant proposal tomorrow, which you maybe are. What would you have written into that?
Our most recent grant proposals devoted to medical diagnostics. So we look at nucleosome repositioning in specific cancers. Our technology is generic, so it can be applied to any type of cancer, or we are also interested in other medical conditions such as endometriosis, for example. But so, of course, every medical condition has its own peculiarities. So our current interest is you look at a specific cancer type, and then you find what are the specific features of nucleosome repositioning in that cancer, which you can use for early cancer diagnostics and for other types of analysis like stratification, monitoring of patients.
So is this liquid biopsy approach, is this something that is in the clinic already, or is this something that is still in development, or is it like cheap enough or affordable enough that it can be used in the clinics?
Liquid biopsies, they had several generations, so to say. The first generation of liquid biopsies, which was based on mutations, it has been in the clinic already for at least a couple of decades. For example, it was used in prenatal testing quite often. So that is already well established, and many companies. This is already a multi-billion market. And the second generation is based on DNA methylation, and this is already also either in the clinical trials or even entering the clinic. And the third generation, this is what we are developing based on nucleosomics. This is, well, the next, we believe, the next step.
Okay, I see. So for the last 36 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
Well, if our podcast goes live quite quickly, and then there is one timely thing to announce, that we currently have openings for 2 postdoctoral positions. And specifically, I would like to invite applications for a person who has knowledge of both machine learning and bioinformatics as applied to genomics. I know it's a unique combination of skills, and also interested to work with us and excited about our research. And so such people are very much welcome to email me, and we would be happy to consider.
I will make sure to put a link in the show notes so that the contact is easy. So you are also, and this is not from the top of my head, because you are also involved in many different outreach projects. What is your motivation behind that? Because you were like also involved in the early stages of the fragile nucleosome, right, and things like that. So how did you get into that, and why did? Yeah, is that something you want to explore further?
Well, it started during the pandemic. The fragile nucleosome was probably the first series of seminars, and it was one can definitely say it. If it's not the largest, one of the largest such communities in our area, and people were actually very happy that this existed. It's also, it's not just seminars. It's a forum where it's a mentorship program. So it's a very, very good thing to do. And the forums are still active, where people are helping each other with articles and questions. I mean, isn't it cool?
Yeah, definitely.
And I'm also involved. I've been involved in a similar activity for the International Nucleosome Consortium. This is the European initiative, and also in the Biophysical Society, which is US based, we have a subgroup called Multiscale Genome Organization. And I'm also involved there with similar type of activities. So these are all in our area, nucleosome positioning, maybe going beyond nucleosome positioning as well. So chromatin in general, gene regulation, one can say. I mean, I think it's important that people have this possibility to interact, and it's cool.
Yeah. Yeah, it's a great thing, yeah. So to finish, what is your personal definition of epigenetics?
Well, I liked the definition of Mark Petashny, where transcription factors were also included in it. I'm still a bit of I'm a transcription factor person as well. So I like transcription factors not to be thrown away entirely, but I love the methylation as well. So.
Okay, I see. So thank you, Vladimir, for your time and for being on the show.
Thank you very much. Thank you. All the best. Thank you.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.